Hello, we're glad you joined us for this live webinar, Recent Progress in Cellular Medicine with an Eye to Precision Medicine. I am Christina Mahalik of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars. To learn more, visit labroots.com. Let's get started. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click on your green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking the green Q&A button at the lower left of the screen. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. It is my pleasure to now present today's speaker, Giacinto Scoles, PhD, Donner Professor of Science and Professor of Chemistry Emeritus. Giacinto Scoles is Donner Professor of Science Emeritus at Princeton University, Distinguished Adjunct Professor in the Department of Physics and Biology at Temple University, and Distinguished Scientist at Synchrotron Electra in Trieste. He is presently Senior Grantee of the ERC in the Faculty of Medicine at the University of Udine, Italy. His career has spanned an unusually long length of time and an equally unusually broad range of subjects. He has developed instrumentation in mass spectroscopy, gas viscosimetry, cross-molecular beam scattering, clusters formation and spectroscopy, surface monolayer, grazing incident X-ray scattering, superfluid helium droplets, manipulation of biomolecules, biomolecular interaction at the nanoscale, and medical nanodiagnostics. He and a group of collaborators have recently patented a method to count circulating tumor cells based on the measurement of the metabolism of every cell isolated in a droplet of blood of several picoliters. When the droplets results to become acid, that indicates the presence of cancer cells. He has received several recognitions: the Hirschfeld Prize, the Gold Medal of Honor of Italian Chemical Society, the Ben Franklin Medal for Physics, 2 honorary doctorates, the Dubai Prize of the ACS, the Pilot Prize of the APS, and the Lippincott Award of the OSA. He is a fellow of the APS, OSA, and the Royal Society of the UK, and the Royal Science Academy of the Netherlands. He has published about 300 papers in peer-reviewed journals and has been quoted by his colleagues approximately 19,000 times. His H-index is 73, while the I-10 index is 224. He has supervised about 100 PhD students, an unusually high percentage of which now hold positions of responsibility in academia and research. It is my pleasure to introduce Professor Scoles. Professor Scoles, the floor is yours.
Thank you for your kind introduction. Now, let me go to the first slide, please. the talk will be about recent progress on single-cell medicine with an eye on precision medicine of cancer. Just a second. The mission statement of Precision Medicine Initiative reads, to enable a new era of medicine through research, technology, and policies, and empower patients, researchers, providers to work together towards development of individualized treatment. The statement smells like politics from very far. However, it's useful to take it as a starting point to research and to reason about the subject. The idea came, but not exclusively from the successes of analytical biochemistry, mainly due to the progress of nanotechnology, leading to single-cell analysis and other factors like this. Precision medicine and personalized medicine have been two terms that have been used interchangeably so far. However, I propose to grab the chance to the existence of two terms and define the same thing to distinguish between them among the two. Really, personalized cancer medicine can be taken to task because certainly it will be very expensive. If it will work, it will be very expensive. To wipe out the advantages of efficient drug use, which is a classic of nanotechnology, using less amount of drugs, so the drugs will be less expensive. But personalizing the treatment, it will be certainly more expense involved, therefore wiping out the advantage. On the other hand, instead, precision medicine, instead of having the ambition of solving the problem of each patient, will have the ambition of solving the problem of a stratified group of patients. For instance, a phenotype in common to, or say, 100 patients. Suppose you have a cohort of patients of 1,000, you can sort out the phenotype of 900 or 800 or 7, whatever number of them have a common phenotype. You can address that, take the cells from those patients, treat them, and if the treatment is okay, you can have the ambition to treat the patients also after that. In that way, the phenotype is a less sure way to cure the individuals, of course. There will be only a chance the individuals are cured. For instance, out of 1,000 patients, you can have a phenotype common about 100. You make a cure for these 100. It will not work for all 100, but maybe for 90 only, because there will be subtle differences later on, however, not so subtle, but they determine also the outcome of the result of the cure. For instance, you can have changes in the DNA, subtle changes in the DNA that are however, important for the thickness, and that therefore environmental changes on the patients make a different outcome. We will have to postpone that subtle differences to later. If we can cure 90% of patients of a certain phenotype, it will be a great success for the method. Therefore, precision medicine will be successful. But before going to have the guarantee that we can cure 100% of each phenotype, there will be some time research later on necessary. During the rest of my talk, I'm going to give two examples of what I'm talking about so far. One, taking the CTCs, circulating cells, tumor cells in the peripheral blood, and counting them, and repeatedly, and then increase or decrease will be a function of, it's been demonstrated already in the literature, a function of the progress in the cure of the sickness. Therefore, we can use this counting circulating in the peripheral blood as being useful to monitor therapy. Second example will be actually going into these cells and try to get the phenotype common for the stratification of patients. We started from one type of cancer, the breast tumor cancer, and then go into that. We only sample a small number of sample patients. It will be about something like 56 or 60, because it's very slow method, going very deep. But I will indicate later on how the method will be speeded up eventually in a short time from now. But so far, the work has been done slowly with the DEPARAY machine. It's a machine that treats cells separately, and therefore it has taken time to get to the demonstration that the cells are CTCs, so circulating tumor cells. So these two examples will be very useful to understand what I mean by curing a phenotype instead of curing a person individual. So far it's too ambitious, in my opinion. That's why I make a distinction between personalized and precision medicine. I'm interested in having feedback on this distinction. I don't think it's been made many times in the literature before. Okay. Now, let's discuss cancer treatment today, how it is. Diagnostic link. You go to a metastatic tumor cancer, for instance, and you start this therapy cycle. Just a moment. I've lost. Okay. This therapy cycle, we define a timeline when the first chemotherapy cycle starts, and about 1 week after that, you need to wait 8 weeks or so to see the tumor has any progress, so if the therapy is working or not. If you count instead tumor cells in the peripheral blood, the increase or decrease of those can give you an idea if the therapy is working or not. If it's not working, you change it or you discontinue it. A great increase in the quality of life of the patient as a minimum. And if it's working and you have confirmation in 1 week, you can increase the doses or decrease it slightly, depending on allergy coming out and things of this sort. In any case, 8 weeks is a long time to wait if you don't have CTCs. And that's cancer therapy is slow. Breast, colorectal, prostate cancer technology are available everywhere. Quite clearly, diagnostic is a quick link in cancer care, both for screening and early detection of cancer, which is very present in many kinds of cancers, and also in deciding about the progress of the therapies, positive or not. Counting tumor cells can instead being very, very sensitive. It's very difficult because typically if you have to recognize a cell, you have to sorry about this. You have to count them and to find them first. To find them, you have to find like a needle in a haystack basically, because there's 1 in 10 to the 9 cells are cancerous typically. If you include the red blood cells, you exclude those 1 in 10 to the 5 or 1 in 10 to the 6, 1 per million. It's always a very big problem of detection. But it's been shown in the literature already using a machine, Veridex machine, the only machine FDA approval, that if 1 to 10 CTCs per 7.5 centimeters of blood is present, then there's a good prognostic prediction about the survival of these patients. If more than 1 CTC per cubic centimeter in 5 per 7.5 centimeters, then the prediction is poor. So we can have test the resistance of drugs developing, the mutation coming in, all the particularities that cancers have that make life so difficult for the therapeutic agents can be bypassed by this method, because while it is a lifetime, it is a time sensitive for therapy efficiency method. The literature there is in 2004, Cristofanilli was the first in pointing out this prognostic predictivity of the CDCs. And since then, many other authors have agreed to this. In this sense, you can now talk about precision medicine, because you can take that particular class of patients that respond in that way and treat them generally. In this way, if the cells are decreasing in the blood of all patients, you can have good hopes for the future. If they are increasing or they are remaining the same, therefore the prognosis is bad, and you have to change therapy. You can decide that in very few weeks, 1 or 2 weeks, 8 to 10 weeks, which is normal, if you use imaging as a way to detect cancer. Because the gold standard in this field is very direct machine, monitor CTCs using magnetic immunosensing, using antibodies linked to the magnetic nanoparticles. By using EPCAM, they have selected to count only epithelial cancer cells. But we know that because of the epithelial tumor mesenchymal transition, quite a few cells cannot be counted in this way. And I will address this problem in the second part of the talk. Let's go to metastatic tumors. Metastatic tumors are the main origin of death in cancer. They can differ greatly from the primary tumor, and major cause of cancer-related mortality is tumor metastasis. This can change as a consequence of the therapy, can become refractory to therapy. Taking biopsies from metastasis in patients is very difficult, because sometimes metastasis goes to the brain or places like this where there is not accessibility. And therefore, you need slow progress for diagnostic cancer. If you count the CTCs, in my group, I asked the students, Fabio Derbeni and Matteo Soletta, to come up with a method that will be better than what was existing. Which later they proposed to me to use metabolism as a biomarker for cancer, and to my big surprise, no one had thought about it, just that. So we started like very fast work, 4 minutes at a time. It was 2.5 years ago, 3 years ago. Probably it's not been as fast as we thought, but it's been definitely there. So, the Warburg effect is well known. In a cancer cells, the glucose metabolizes into lactic acid, acidifying both the cells and the environment around it. While in a normal cell, the glucose generates ATP, and therefore doesn't acidify the environment. Using this effect, which is very well known, established 60 years ago by Warburg, we can decide if it's well known, for instance, that the environment around the tumor is acidic, but you cannot take changes in that, measure changes in that type of acidity because it's too small. The blood flow dilutes the effect and you don't see big changes of acidity. But if you take any cell, cancer cell, you isolate it into a droplet of water, and then you flow it, and you expose it to sugar. The metabolism of that sugar will be making the cell more acidic. The proton pump will pump the proton out, so the cell to survive, and the environment around the cell will become acidic. Depending how big the environment is, when you take a cell of 10 picoliters, it will become smaller, changes in the acidity than if you take a cell in 1 picoliter. But the volume of the cell is much less than a picoliter, and therefore you can take a droplet 10 times or 100 times the volume of the cell, and then measure the acidity in that. So you do the following. I feel I need to tell you that I have blepharospasm. I have a sickness that my eyes close from time to time, and I have to touch them to open them. Sorry about this. Okay. The working principle of our machine is to detect pH changes in the extracellular environment at a single cell level. So you encapsulate every single cell in a droplet, and then there you see a droplet containing a cell of about 20 microns diameter in a 50 micron droplet cell. That is a volume which is 10 times or maybe 100 times larger, and however small enough to detect the big change in pH due to the acidification of the Warburg effect. So a comparison here with the Veridex machine in our method. The Veridex machine, you look for protein expression at the surface of cells, and we look for metabolic differences in picodroplet. Spectrum is very narrow, epithelial cell only. You can look at all very broad spectrum. Every cancer cell is known becomes acidic. Very few exceptions to that. Cell isolation is not possible because the antibody, the magnetic particle, fixes the cell and kills them basically, and we enable our method enables cell alive and label-free to be used for genetic purposes after that. The cost is €500 analysis for using the Veridex, and a reduction of more than 10 factors of 10 for our method. These are details. It is important because we start with a preservation of blood. You put the blood into a microfluidic environment with a microscope and a laser. I don't see the droplet moving here, but I've been told you may see the droplet moving. In any case, you can imagine that the oil is flowing, the sample is flowing, they merge, and this microemulsion is being formed. Then the droplet moves down. Incubation time is 10 minutes at 37 centigrades. And the droplets are collected at the end. As you see in this example, several droplets have been collected. One of them contains a cancerous cells, and the red arrow there, and the fluorescence coming out of there because it means that the environment is acidified, while the others are all dark. Even this one where there is 3 cells, it will affect the acidity of the droplet. You see here a pump, very normal, low pressure pump in the fluidic circuit, and here you see the droplet moving again. They move down. You can make the droplet go very fast. You can form more than 1,000 per second of droplets. And you can look in a millisecond, it's all of them. According to this circuit, you have a laser sending light down, going to the sample, and we use a ratio method, 2 peaks, that if one goes up, the other down, the cell is acidic, they don't change, The cell is not acidic, they remain as they are. We calibrate first the machine by having these peaks in the control environment where we change the pH of the water droplets coming into the machine without any cells, controlling well the pH droplet. And you can see there some two peaks. It's a ratiometric method. The two peak changes, and here this change increases as the acidity of the droplet increases. And the calibration curve is very good. You can see there on the left high side of the slide. And we can take cell lines tumor-derived, which is not yet a cancer cell. It's a cancer cell, but it's from a cell culture. We can spike into the blood and see if we can detect those at least. And indeed, here you see a demonstration of the technical work. Okay, here there is a sample of white blood cell only. You don't see anything there. And if you add breast cancer cell line, MDA, MB, 231, you see plenty of points where the acidity increases. On the axis, x-axis is the size of the droplet, but it's irrelevant. You can take any size there. And there is, in typical here in the center of the slide, lots of cancer cells. And here the results, the two peaks as they appear. And you can take MDA, MB, 453 breast, metastatic carcinoma, and you have plenty of results there. HT, colorectal cancer, 29, adenocarcinoma, and again, spike 1% or so in the blood, 1 per mil, even less. And you can detect them quite easily. Sometimes you see some stratification of white blood cells, and we talk about this perhaps later on during the talk. To see how much time response of the machine is, you expose the pH changes to the sugar for a different amount of minutes. We typically find out 10 minutes are sufficient. Otherwise, if you go into asymptotic region, it's not useful longer. You only add to the time of the analysis. Therefore, 10 minutes are sufficient. Even less probably when the machine will be optimized. Spike samples now. We take all blood and we spike in MDA, MB, 468 cell culture, and you can see there we have plenty of signal acidification. When there's lots of cells that are moved, because lots of droplets don't contain any cells. And this is a verification that biologists do. Sometime happens that impurities or something, the droplet changes pH in time, but then if there is no cell in it, we examine every droplet with a machine, photographic machine. We know that is a pure result and we can eliminate it. Then we have healthy donor of lymph blood, and there is no results, no acidification droplets, and the AB 468 spike, and there is results. So this really is the first time that we can see that we can get results, because healthy donor has no signals, while spiked blood of healthy donor has signal. If you do patient samples, healthy donor lymph blood, no signal, MDA, spike, and colorectal metastatic patient, we have signal, many droplets were change them, the acidity. Typically we get too many droplets of the acidity with respect to the Veridex machine, But that's a different result that we are achieving right as we speak, and we have to discuss it separately. Mesenchymal transition is always present, and we can see that we don't lose the signal. Well, if the mesenchymal markers are on the cell, they are missed from epithelial transition, and therefore cannot be detected with Vedics. So we should always see more the Vedic sees, or the same. In this result, we can discriminate between cancer cells and white blood cells. Despite result, we can detect cancer cells in blood, cancer cells lying in blood. As a result, we have a sample, see a pattern for a cancer cell line in a patient. This is all slide. These my students and co-workers, they recently quite a big progress, but they're not like to speak in public about it. They've been conducting a comparison with Vedics, and we need now to sort the droplet out, because now we have a method to make many droplets, many cells, and have those be assigned to study resistance to therapy and things of this sort, so we can make genetic profiles of the tumors, and then they change it in time, and they change it, the heterogeneity of the tumor itself, and think of this sort we can all study. Characterize the source itself, phenotype and genotype, by the I will say. In particular, we need to compare our results with those of the Vedic apparatus. Expect differences, but consistent in the case of doing measurements. It means that you should have always the same number of cancer cells or more than the Vedic machine. We have developed this method with Professor Hook at Warburg University in Nijmegen in Holland, and his Professor Aiger Spiruska. And people in my group are Matteo Suretta, Hans Cilotto, Daniele Cesselli, Alfonso Combatti, Fabio Del Ben, and Michele Bolfoni. And this is about Michele Bolfoni's work that I'm going to discuss with you next. Characterizing CTCs. We could not do it with the same level of separation that we have achieved now using the droplet method, because then we have not sorted them, droplet out. The sorting experiments are starting now, and a new circulating machine, microfluidic machine is being prepared for sorting. But if you use a very slow method, you can probe deep and study 130 patients in 1 year, for instance. Of course, a lot of them. There's also time for this. But you can then get an idea about if the method is going to work when you will do it faster. And the results we are going to discuss now, obtained by Michele Bolfoni, are the following type. They can detect and count the CTCs. A sort, contrary to what we cannot do, using the array, each cell, and they sorted without damaging it. They characterize it and clinically validate their method. So it takes for them to arrive to a characterization of a cell 1 day. So you can see in this way that a patient in 2 weeks have to do a patient or so, and do some in parallel. 1 year of work is 50 patients, which is a lot of work for arriving to the result that they show you, but it's possible. Then they can take epithelial markers, mesenchymal markers, CD45 and hooks. Cells are being considered viable in the depth array, don't damage them, and they separate them. And I show you the result next. Here you have Okay. That peak coloration of the nuclei opens all of them, of course. Then there is mesenchymal markers, the red ones, epithelial markers, green ones, CD45 marker, and the so overlap of all of them. As you can see, there are cells which are both mesenchymal and epithelial markers in them. And how they will react in a separate machine, they can easily identifiable. While in the very machine, they are not clear what result they can do. So, by analyzing this cell in detail in this way, Michela Buffoni has come to the same conclusions. She is compared with the search machine, and then she has associated between the clinical pathological features of the CTCs and the distribution of CTCs for populations. For instance, I will show you in the next slide that HER2 positive diseases, breast cancer, has found lower number of mesenchymal cells. In this case, the counting of the cells will be in agreement with the Warburg machine. Bone metastasis, higher number of epithelial cells, higher percent of the cells. Again, here is a good case for comparing with the Warburg machine. In liver, all the cases where the comparison with the Warburg machine is good, listing higher number of epithelial cells, lower number of mesenchymal cells. Imagine a higher number of epithelials, number of localization of the site for greater than 1, therapeutic line, greater than 2. Imagine the higher number of negative cells for both epithelial and markers. What she has found was unexpected to her, and to us all, the fact that if the cells are in transitions between epithelial and mesenchymal, show markers for both types of cells, and there's a lot of these cells, and the prognosis of the patient is very poor. That's a big clear diagnostic procedure that she has introduced. It's not possible with the Warburg machine because the mesenchymal cells are not visible at all. Okay. For instance, percentage with mesenchymal markers, if it is more than 50%, the survival rate is very good. While if larger than 50%, survival rate becomes very poor. Quite clear distinction here between survival rates, as you can see. So, using the array, it is possible to identify and sort live mesenchymal-like and epithelial-like disease as well as mesenchymal cells. So a single cell can undergo downstream analysis. Disease population could help in estimating metastasization pattern and outcome because of their association between clinical pathological features. Results need to be confirmed through larger numbers, larger trials, of course. The fraction of epithelial cell expressing mesenchymal markers seems to be correlated with survival. Number of disease expressing mesenchymal markers is to be prognostic factor both for survival and progression-free survival. By, say, low number of mesenchymal markers correlated with good prognostic factors. progress now is the direct comparison with research. 10 patients have been already carried out, 22 are ongoing, and then new generation sequencing of single cell is being carried out to study resistance to drug and similar things. By that clinical result, number of patients need to be increased from 160 to 130, and a grant has been obtained recently to extend it and like to culture cells. These co-workers for this type of work, Daniele Cesselli, the leader of the group, with Professor Beltramis, Carla Di Loreto, Stefano Manzinotto, Mario Sorrentino, especially Michela Buffoni, carried the bulk of the work. Statistical support, Mini Misura, Padua, where the comparison with the cell search machine is being done, Rita Zamarchi, Elisabetta Rossi, our technology group is our group, and Silicon Biosystem Manuela Brandi, and Peruzzi, and Giuseppe Nicola. Genomics Michele Morganti, the big institute of genomics at Udine, where this group is located. Thank my wife, gives me great support because of my age. It's not easy, as you can see, give a talk of this type without being taken care of. And thank you to all of you.
Thank you, Professor Scoles, for your informative presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Professor Scoles will answer as many questions as time permits. All right, let's hop right in. The first question today, Professor Scoles asks, in your opinion, is the distinction between personalized and precision medicine is necessary?
That's why I introduced it. It is essential to make the system work for stratification of patients where 100 or 10 patients are on the same phenotype can be treated similarly and therefore have a hope to survive together. Why really personal medicine is far away and I don't want the failure to do real personal medicine come into the way of this precision medicine which I introduced it for phenotypes and genotypes common. That I think it's possible. But of course we have to prove it now with the facts. But that's my personal opinion, that's why I make the distinction.
Thank you for your answer. Our second question asks, is the metabolism of a valid criteria to locate and sort CTC cells?
And this was known in a sense without our machine that counts cells one by one. Metabolism has been a criterion to study cancer already in bulk, but eventually doing tumor cells, and by seeing that for every type of cancer we have tested so far, tumor cells respond acidifying the environment and being metabolically responsible for what they do. It is a good method to pursue for any type of cancer so far we have found. Yes.
Thank you. It appears that we have one last question for today. This question asks, can metabolism be useful in studying or practicing cell medicine in other illnesses?
Yes. We hope very much so that this is the case, and we have a couple of trials feeling around to study, extend the study of metabolism to other type of cells, other type of sickness. For instance, we are convinced that a difficult to diagnose illness, typically the rheumatic arthritis, can put in circulation cells that, if provoked, will change metabolism is altered. Everybody knows that, but if we analyze these cells one by one, with a certain percentage of them, we show features that can be taken proportional to the diagnostic of the sickness. And if that's the case, major progress will be done in this case because there are no good diagnostics criteria, quantitative criteria for the type of sickness. It's normally judged only on the symptoms. If you have pain and your joints deform, then you have rheumatology. And then, but there's no, if you get the medicine, it's the way the pain disappears before you know if the medicine is working. But if the number of cells decreases, then you have a method. We are studying this type of work at present. We are very preliminary stage at the proposals and things of this sort. But I think it's a good method, metabolism also in this case. Thank you.
As we don't have any further questions, Professor Scoles, thank you for your time today. Would you like to leave us with any final comments?
I thank you for the opportunity to present this work, and that's fine. I don't have any further remarks to do. Thank you.
I would like to once again thank Professor Scoles for his presentation. I'd like to thank the audience for joining us today and thank LabRoots for today's educational webcast. This webcast can be viewed on demand through May 2017. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. I'll see you next time here at labroots.com. Thank you and goodbye.